Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show. Today, bringing you a rather really awesome guest involved in science and technology that's creating a better tomorrow for all of us. Today, we have the honor of being joined by Dr. Jackie Kunzler, who is the Senior Vice President of Research and Development, as well as a member of the Executive Management Committee at Terumo Blood and Cell Technologies. And there, she focuses on innovation and development, leading the way for unlocking, as they say, the potential of blood and blood therapies, cell collections in various sectors, including blood banking, plasma-based therapies, as well as various cell and gene therapies. Dr. Kunzler joined Terumo from Baxter Healthcare, where she held successive leadership roles in their business, including as Baxter Healthcare Senior Vice President for Quality and Regulatory and Head of Global Life Sciences. Dr. Kunzler holds a Ph.D. in cell biology and neuroscience from the University of Illinois. She did her master's degree in toxicology at the University of Florida, bachelor's degree in biology and medical technology and focus on lab medicine, University of Iowa, and she did her MBA in healthcare administration from Benedictine University. A lot of really cool topics to get into today as we fully explore this concept of unlocking the potential blood, as they say at Terumo. Honored to have with us Dr. Jackie Kunzler. So thank you so much for taking time to come on the show.
Well, hi, Ira. Thank you so much for thinking of me and for inviting me to be on the show. I'm really excited to have a conversation with you and look forward to going wherever it goes. So let's jump right into it.
Absolutely. As I mentioned before the show, I mean, this is an area that we touched on a little bit, probably not as much as we should, because an extremely important set of technologies and topics. Before we get into all of that, would love to start off, by handing you the floor for a little bit, just to talk a little bit more about you. You know, one of the fascinating things in sort of looking at your bio and sort of your career arc was that right after your bachelor's degree, you sort of went fast and furious into industry, and a lot of what I read in terms of your education you did while working in terms of your master's in toxicology, your PhD, all this while sort of learning the ropes inside a major healthcare business like Baxter. Talk a little bit about yourself, a little bit of your background, and sort of what got you focused initially on med tech, on lab medicine, as well as healthcare admin.
Sure. So and hopefully I I don't go into too much painful detail for you, but even from when I was young, I was my best subject and favorite thing was science. I was very fortunate in high school to be given the opportunity to pick any field that I would like to do an internship in freshman year of high school. I picked laboratory medicine and went and worked in a doctor's lab for a month, roughly a month. Fell in love with it. From there, I decided to become a medical technologist, which is laboratory medicine in a hospital setting typically. And actually, I did go work in a hospital, Elmhurst Hospital, for 4 years right out of college. I was a clinical chemist in the chemistry laboratory, and that's doing things like pregnancy testing, drugs of abuse testing, cholesterol testing, all that type of work for patients in the hospital. At that time, I also got married, and one of my big dreams was to have a big family. So I decided to start a family with my husband. And at the same time, I recognized that the real path forward to a career in a hospital is hospital administration. So I started the family. I went back to school, got my MBA in healthcare administration. And during those 4 years, I had a lot of conversations with some of my bosses and realized what I really wanted to do. Well, two things. One was raising a family and working in a hospital. You have to work all the holidays, the weekends.
Mm-hmm.
It's a really challenging situation for raising a young family. And so I naively thought, I want to cure cancer, not not realizing at the
That's
time
cool.
what the size of that ambition, right? So I decided that I needed to get into a research setting. And strangely enough, my boss's boss at the time in the hospital was interviewing at Baxter for to be the head of clinical pathology. And that's typically where med techs report in.
Mm-hmm.
So he was kind enough to take my resume with him to his interview. And in the end, I ended up getting hired and he didn't. So he stayed. Crazy situation, but he was happy for me. So that's how I started at Baxter. And I started at Baxter as a med tech.
Yep.
Same kind of role, but doing testing on employees for health screens as well as we had a preclinical facility at the time. So we did a lot of animal studies. And so you still use clinical chemistry and hematology for that. And from there, one thing led to another and different roles and, taking opportunities as they came and looking for bigger opportunities. I realized I didn't have enough science background. That's how I felt.
Mm-hmm.
So I looked for, and by now I had 3 kids. They actually hired me 8 months pregnant with my third baby under 3. So that was really a great opportunity. And so I looked for a program I could do online, and I found a toxicology program at the University of Florida, Gainesville, that was all online until the last week where you had to go defend your knowledge. And so I enrolled in that. I would do all the work after the kids went to bed and after work. And then serendipitously, which is oftentimes exactly what happens, the toxicologists at Baxter went their separate ways. And so I graduated like in August, and in September, I became the head of toxicology. And it never was what I planned. I had no ambition to be the head of toxicology, but it just happened that way. And then I really wanted more career advancement, and basically I was told I needed a PhD.
Mm-hmm.
And so I was, and I was surrounded by people who didn't have PhDs, but for some reason I needed one. So I went back to school. I convinced the University of Illinois, a really great dean there, to give me a shot at working full time and getting my PhD, which is not the conventional way to do it. Typically, you have to give up your career and go full time.
Yeah.
So they did that as a pilot program and it worked out. And so at the age of like 45, I finished my PhD in neuroscience essentially. And from there, things really took off. At that time, I was leading the life sciences organization. I decided to take a leap outside of R&D and try my hand at marketing. Again, I was very fortunate to be given an opportunity to be part of a new organization at Baxter that was called Business Model Innovation. And we worked largely in India and China,
Mm-hmm.
trying to bring Baxter's products to emerging markets. And I was having the time of my life, quite honestly, doing that when Baxter found themselves in need of a person to run compliance. We had some regulatory issues that arose, and so they asked me if I would leave marketing. That was an international marketing role, and become the head of compliance, which I did. We started turning all the regulatory issues around, and then a new CEO came in, and I was hired to be the head of quality on the leadership team. So, a crazy route all over the place for different reasons. And I was there for 30 years. I retired about a year and a half ago, and I really was looking for an opportunity to lead an R&D organization. And I was incredibly fortunate that Antoinette, Gavin, and the Terumo BCT team gave me that opportunity. And so for a year and a half, I have been leading the R&D organization out of Denver.
Mm-hmm.
My family still lives in Chicago,
Yeah.
and I'm working working in Denver and commuting. So
No, it's an extremely impressive journey, and I enjoy thinking about all those steps. Some of them accidental, but nonetheless, I mean, it's
yeah.
this is where it's gotten you. So no, it's very impressive. And what one thing I wanted to touch on, Jack, before we get to Terumo in a couple of minutes, I would just like to stop at your PhD for a moment in 2014 because the topic of your PhD was this interesting, interaction between amyloid beta, which we all know a lot about in terms of its connection to Alzheimer's, how estrogen may be kind of beneficial there, but how this other apolipoprotein E, that if there's too much of that, that interferes with the estrogen, and there's an interesting little cycle there. Aside from it just being a cool topic, I think we're going to touch on something a little later on when we get into the blood therapies in terms of, when we address these complex diseases, there's not just one thing going on. A lot of drug companies have focused on amyloid beta for quite a while now. We've not made much progress. I wonder if you just say a couple words about this little body of work, because I think we'll touch on it a little bit, how, some of the things you're doing in terms of, blood therapy and apheresis and so forth that allows you to modulate sort of combinations of bioactive substances in our bodies. Really is an interesting paradigm beyond, how we typically think of treating disease. So say a couple words about your PhD, if you would.
Sure. So let me just start by saying why I ended up in that particular lab and what drove me to the dissertation that I ended up with. First, my husband's family has Alzheimer's disease in the family. And so we were at that time watching his grandfather struggle through that disease, and who knows where else it'll show up. So I had a keen interest in that. I personally was on hormones. And so the idea of hormone replacement therapy after menopause was really interesting to me,
Yep.
independently of the Alzheimer's disease. And the individual who was doing Alzheimer's research at the time was very focused on the interaction of amyloid beta and apolipoprotein E.
Mm-hmm.
Apolipoprotein E has three variants, E2, E3, and E4.
Yep.
E4 is associated with Alzheimer's incidence, let's say. Obviously, the plaques in the brain have amyloid beta in them. And so in my mind, I was thinking, what kind of a study could I design to look at all of this and how it's interacting? And so what I ended up doing, and let me just disclaim that this is a very small study in mice, but that lab had genetically engineered mice to express different ApoE lipoprotein variants and to develop plaques. And so I took female mice, I put them into menopause by removing their ovaries, and then I divided them into cohorts that either received hormone replacement therapy or didn't
Hmm.
by apolipoprotein E variant. And so, many combinations. In the end, I took out their brains. I looked for total quantity of amyloid beta as well as plaques. One half of the brain would be quantitative, the other would be plaque pathology imaging. And what we found was hormone replacement therapy was beneficial generally in ApoE2 and E3, but
Hmm.
not in E4.
Hmm.
And so that would seem to imply that ApoE4 is, a stronger indicator or contributor toward the formation of Alzheimer's disease.
Hmm.
So that was the study. I thought it was pretty cool, to be honest, and it addressed, everything that I was interested in. So
Hmm.
it worked out well.
Yeah, no, it's extremely cool. And again, in a little bit we'll touch on sort of the combinatorial aspect of, let's say controlling things in the blood and how they're, they really offer some interesting modalities. But I wanted to put that out there as well so the audience knew some of this area of research that you were also involved in. So I appreciate that. Yeah, let's introduce the organization a bit because you lead R&D at Terumo Blood Cell Technologies, which is part of the broader Terumo organization, which I'm a pharmacist by undergraduate training, and in the pharmacy, I still remember the huge wall of Terumo insulin syringes and other products like that. Um, just talk about the Terumo organization for a couple minutes, if you would, and just a little bit of BCT. You mentioned you're in Denver. Just talk about where the organization sort of resides, and then we'll get into its technologies and various offerings.
So Terumo, as you said, Terumo BCT is headquartered in Denver. We do have R&D sites elsewhere, India, Costa Rica, and other places. But we are one, if you think of it as one division of a larger parent company that's called Terumo. And Terumo originated and resides in Japan, in Tokyo. And Terumo, like I said, has multiple divisions that look at cardiovascular uh, equipment, solutions. We have in California an acquisition that is focused on neurology, and we make hundreds and hundreds between all of the divisions. We make hundreds and hundreds of devices used for treating blocked arteries, for treating aneurysms, for treating surgeries. And then Terumo BCT, the BCT part of Terumo is Blood and Cell Technologies. And so, as you said at the beginning, we are very much focused on the importance of blood. And we make primarily instruments that can remove different components of the blood for a wide variety of treatment purposes. So that's kind of Terumo in a nutshell.
Hmm. Hmm. And while we're on that, and we'll get into technologies in a minute. You again, sort of restating this, unlocking the potential of blood, as it says on your website, to do more for patients tomorrow than it does today. And I'd love to have just a couple minute discussion about, and this is an odd little question, I think be kind of obvious, but it's really not. I think. what is blood? We're taught. We were joked about this when we were chatting before the show. We learn about our white blood cells and our red blood cells and our platelets and our plasma, and that's as our high school, I mean, college biology, pretty much. But what do we know today? We know that there are literally thousands of bioactive substances in the plasma. We know I don't know how many types of T cells that we've identified to date. Different types of platelets. I mean, it really is the ultimate sort of combinatorial bioactive substance. When you're just thinking about, what do you think about when you think about blood from a not just personal perspective, but also just from the Terumo perspective? I mean, it's more than just this red stuff that, that floats around in us.
You reminded me, you flashed for me in my mind. I don't know if you've ever seen the biochemical pathways poster.
Oh yeah,
It's this poster that has every biochemical pathway in the body diagrammed, and how they all relate to each other. And it just blows your mind to look at it. But, you know, blood is survival. Blood is everything to the body, to the tissues, to the organs. It's full of lots of good things, and it certainly holds the key to understanding when bad things are going on as well. And, the beauty of working in science is studying diseases, and trying to figure out it's like solving a puzzle every single day. And it all starts with the cell. The cell is, the single unit that starts everything. And in the blood, like you said, there's blood cells, red and white. There's plasma, there's platelets, there's tons of things like hormones, there's proteins, there's signaling molecules, there's things like A beta, amyloid beta. There's just an infinite number of things that give you information about what's going on throughout your body. And so, I mean, it's incredible, probably 30 or 40 years ago, people thought we knew everything about blood. And the more you study it and the deeper you get, you just continue to find, smaller and smaller molecules that mean something. And so it's a really exciting place to be and to work because every day is different. And there's certainly a recognition that although we've learned a lot and we know a lot, there's just so much more constantly to figure out. So if you like solving puzzles, it's a wonderful place to be and to work.
And it seems like, in that analogy in solving puzzles, right? Um, there's not one way, let's say, to do what you do. And when I say do what you do, it's at the core of, a major part of your business is this principle of therapeutic apheresis, basically this procedure that allows you to ultimately isolate, remove certain abnormal blood components that ultimately could be causing disease to treat. If we start from this perspective of isolating and removing, and I know you've referred to this as sort of the bad guys in a presentation that you gave before. Clearly, when you're looking at bad guys in the blood, the thing you do, the magic that gets a sickle cell out of the blood is going to be quite different than some cytokine where I got to get this protein out, or if I took too much whatever, I've been poisoned, and you got to get some small molecule out. That's another set. Talk a little bit about what R&D means in your organization in terms of therapeutic apheresis and sort of the scope of what you focus on a daily basis. Because clearly, again, while maybe some of the machinery might be the same, the details behind each of these possible thousands of things is slightly different. So take a little time to talk about the R&D that goes on in the R&D organization when it comes to thinking about the bad guys.
Yeah, so one of the things that popped in my head, sorry, I'm probably a little bit all over the place, but so much
Oh,
comes at the same time. It's like, how do you sort it all out? One of the really interesting and tricky things, and when I think about Alzheimer's in particular and A beta, initially, if you went back probably 10 years, the thinking was if you just get all the A beta out of the body, you'll solve the problem. And I think this applies to everything that you look at removing. It's there for a reason. So most things are there for a reason. It's not typically, at least in my experience, going all out to completely remove something like a molecule isn't always the best approach, because although it can go awry in some cases, it probably serves a function. And so that's kind of a delicate balance when you're thinking about all of this.
Yeah.
But in our case, what we are capable, I mean, we do a lot of different things. We have the ability to, with our RECA machine, pull out plasma in large volumes, which then goes on to be manufactured into things like albumin, factor 8, factor 9, therapeutic proteins. So that's a commercial application that really is more about, it's not to be donated to patients real time. It's to take all the good guys and make products out of them for people who don't have them. Then we have applications that can pull out very specific parts of the blood system, maybe red cells, maybe platelets, maybe white cells, and use those for therapeutic purposes. So an example might be sickle cell. You could take out sickle cell red cells and you could replace them with healthy red cells. You could take out white cells. You could put them through, I'm sure you've heard about CAR T therapy.
Sure.
Put them into the CAR T manufacturing process and make a treatment for leukemia. You can take out, and I guess if I think about the future, some of the things that we're looking at are, can we selectively remove bad guys? So let's say we think A beta in a patient is a bad guy. Can we put something on a column or something on our machine that will selectively target a bad guy? And when I think about that, my mind almost explodes because there's so many possible applications for that. Typically, in our external world, we think of solving everything with a drug, but there are other ways to do it, to your point earlier. And so, my brain goes, wow, Lou Gehrig's disease, all these other diseases that haven't been solved. Could we come up with applications for those? So that's where I get really excited. But in addition to that, some of the things we have to think about are the rest of the world. As I was doing a few years ago, emerging markets, how can we make our platforms and our products so that they can be transported easily? When I was in the business model innovation role, we were looking at hemophilia in India. And one of the problems, aside from the fact that no one seemed to be aware that there was a cure, the bigger problem was access. And how could we get access to people who live in small towns and rural areas who have this problem? And it's the same for things like sickle cell. When you think about Africa, there's a huge sickle cell population that's untreated. How can we make our products mobile enough, lightweight enough, transferable enough, and at the same time educate people and try to bring the therapies to them? So I would say those two areas. And then the other thing that we work on is how can we be more efficient? So, one of the challenges we're going to have as a society is as the baby boomers get older and older, that population of people who have been wonderfully generous with donating platelets, plasma, whole blood, they're getting older to the point where, they'll be out of that system. And we have a real gap in the younger populations in terms of donations. So, and part of the reason is because it takes too long. So as we look at our R&D projects, a lot of them also have to do with how can we make these collections faster, more efficient, requiring less of a commitment from people. So all of that is kind of, where we're at right now.
Mm-hmm. Mm-hmm. Yeah, I don't always think about the time issue when I'm giving blood, and there's always the plasma pheresis station over there somewhere, and this how long does this take? Oh, we don't have time for that, but yeah, no, I didn't think as much about the that issue, but that's quite interesting. And then as you were mentioning, sort of the different settings and transportability, I know one of the hot topics that I sort of read about online a bit was, how do we, could these technologies, like a lot of things we see in sort of the digital health realm, be made smaller, more portable, and um, you know. Uh, applied to this whole area of health care at home, healthcare in the home and all that, which got me thinking about something like AI, as we're sort of on this digital sort of tech theme, which has been popping up everywhere and sort of every show we've done about healthcare. But I'm just interested in how tools like AI help you in as you develop these technologies to do the isolation, whether big or small. When it comes to capturing or identifying the bad guys or capturing the good guys, clearly, if I'm looking for, hey, I want to only granulocytes and I don't want all this other stuff, or I want some interesting cytokine. I'll just make up interleukin 92. I don't know if that exists or not, but talk a little about AI, machine learning, some of these tools impact your business and allowing you to not just isolate more efficiently, but do things faster and more efficiently.
Yeah, so I'll talk about it in two different ways. Um, One of the areas in our cell therapies, cell and gene therapy business, that we work on is essentially cell culture.
Mm-hmm.
And it's taking out different, whether it's stem cells or whether it's T cells, or it could be anything really. And then part of that process is to culture them and expand them and try to increase the number of them. And that requires a growth curve in essence. I'm trying to make this as simple as possible. To my way of thinking, every single person that goes into that process has a slightly different profile in terms of what's going to make their cells grow the fastest, the best, the most efficiently. And typically we monitor certain molecules like glucose, lactate, that those have to do with, how the cells metabolize in order to grow.
Yep.
And so there's a constant need to balance those types of molecules. And where we need to get to is having sensors that can monitor those real time, as opposed to a scientist or technician having to go in and take a sample and then go run and test it.
Mm-hmm.
If we can get to the point where we have sensors that can monitor that real time and then do a feedback loop, so that those levels of things like glucose and lactate can be adjusted automatically.
Mm-hmm.
That's one area we need to go. And then AI eventually should be able to learn, based on the cell population and what we know about it, what the optimal ratios of glucose and lactate over time would be so that we get the maximum amount of growth in the least amount of time with the least amount of effort from people intervening should be. So that's one way. That requires a huge, in my mind, the best case would be a huge database full of exactly what made this work based on the cell population, based on, underlying conditions from the patient, based on a lot of things. So you could put all that information in and then use AI to generate the perfect protocol to optimize the cell growth. That's one application. Another one that comes to mind because of my time in quality is all companies receive a lot of input from customers. They're often termed complaints. But in essence, it's feedback from a customer. And every time we get that type of feedback, we do an investigation. And the investigations take a lot of time. And many, many times, those investigations or those complaints are the same as we saw 6 months ago, or we saw a year ago, or, things happen repeatedly over time, often. So the application of AI there would be to have a database full of what we've seen in the past. And rather than having to go back every single time and reinvent the wheel and figure out what the root cause was, it could tell us without us even going through that whole very time-consuming effort. So that's another area where I think companies need to go in terms of managing all of the field information that comes in from customers. Does that make sense?
Yeah, absolutely. And it gets me thinking, as one of the things you're outlining here is, Terumo's position in sort of contract development and manufacturing for, many of these, the organizations out there sort of doing the therapies. And I think that's an amazing place to be. And it speaks to sort of what your company's all about. And it got me thinking at the same time as you're talking about sort of, this service angle. I neglected to mention in your bio that one of your other responsibilities is part of the Parental Drug Association, and saw some presentations you gave on this interesting area of extractables and leachables. Thinking again about drug development, okay, obviously there's quality when it comes to making a new drug or protein or whatever, but in your case, you got to think, beyond the machinery and the biologic product per se, sort of everything else that might be happening in this very unique process of taking and isolating and supplying back potentially. Talk a little bit about that side of the quality equation, because we don't usually talk that much about it, but it's kind of a very important piece in this case, in terms of the nature of what this product's about. And I wonder if you could say a couple words about your work in from that perspective, sort of the non-biologic part of things.
Sure. So, the premise, and this goes back to toxicology. The premise in toxicology is that the dose makes the poison.
Right.
That's sort of the tagline, right? And in both Baxter and in Terumo, probably to a little bit lesser extent, but when you're using materials like plastics in particular that come in touch with, in the case of my previous job, it was solutions like saline or dextrose. In this job, it's blood. There's always the potential for things to come out of the plastic into the solution. And so we, as medical products companies, spend a lot of time making sure that there's nothing that's going to come out that would be harmful. And so for about, it seemed like forever, I want to say 8 years, I was part of a committee that worked to write a guidance document on how to test for leachables and extractables, and also what my side of it. There was a chemistry side, analytical chemistry side, that looked at how to extract plastics and things like that. And then there was a tox side that looked at all the chemicals, their structures, what information from a safety perspective was available on different molecules, and to come up with limits, exposure limits, so that people would have a ready reference and there would be a unified approach, especially when dealing with regulators, to knowing and understanding and defending what people are being exposed to in your products. So that's kind of what it's about. It's doing leachables and extractables studies is an extremely involved and lengthy process. And so, our objective was to make the back end faster and easier by providing safe limits.
Mm-hmm.
We do, of course, we do that. We have to do those studies.
Right.
Right now, I would tell you that we're all working toward a new regulation that says non-DEHP in our plastics. By 2030, that's a regulation that's already been approved. Okay. So as we look at alternatives to DEHP, which is a plasticizer that softens plastic, we have to go through all of that, right? And see what comes out with these new molecules and is it safe?
Yeah. It's no, that's quite interesting. Obviously, very timely with all the discussion about microplastics and things like that, and sort of the public sphere. It's again an important piece of the quality equation, and I sort of didn't want that to drop out because we have to think of quality beyond just the actual bio product itself. So no, that's great.
Yeah, and it extends. I'll just add, it's not just the plastic bag, but then there's a port, right? That often has a rubber in it, and that has its own profile. And so it's very complicated when you have an entire bag with a port or multiple ports, and then you have a set attached to it, and you have a needle in the end. And so there's lots of work that has to be done.
Mm-hmm, mm-hmm. Earlier we talked a little bit about amyloid beta, and obviously there's, tremendous potential here to, whether it's isolating, proteins like amyloid beta, a variety of sort of pathogenic antibodies, things that go wrong in autoimmune disease, the hyperlipidemia space, obviously very hot. So there's, thousands of applications here. Sort of there's no end when we get into the area of sort of the chronic degenerative diseases. One thing I wanted to ask about, I thought kind of interesting, would be sort of the area. One of the things besides the cells and the plasma that we don't normally think about that is floating around in us are microbes and sort of the blood microbiome. I spent a couple months ago, we had Craig Venter on the show, and he was talking a little bit about one of these papers he did on the virome that's in the plasma, and we just have thousands of viruses floating around. They're not the pathogenic ones, but they're just hanging out in us, and we don't exactly know what they do. Obviously, our blood has a bunch of pathogenic stuff in it as well. I know Terumo is probably involved in some stuff on this front that may be a little confidential, so we don't have to go there. But love to get your perspective on sort of applications, sort of in the infectious disease realm, and then one of the tools we hear a little bit about is sort of this photopheresis concept, how you can potentially pull blood out, do some type of zapping event, and put it back in. Love to get sort of your broad thoughts on sort of applications in infectious diseases, because clearly we went through a major one the last couple of years, and love to get your thoughts on potential applications on those fronts.
Yeah, so again, a bunch of things pop in my head.
Take your time.
So I'll start with the simplest. If you think about COVID, right? People who were exposed to COVID and recovered became viable donors of plasma, right, because they had the antibodies to COVID. It's called convalescent plasma.
Yep.
Actually, I was one of them.
Nice.
And so, that's one application of a plasmapheresis that can help people. And it doesn't have to be COVID, obviously. It could be,
Sure,
kind of anything. Um, Another one, you mentioned photopheresis. Our Optia machine can be used for something similar. It's for photopheresis. We don't really play in that space directly,
Mhm.
but Optia can be used for that. In essence, you pull out the buffy coat, which is the white cells.
Mhm.
You apply Sorolin to it.
Okay.
You light it up with UV light,
Yeah.
and you put the white cells back into the patient, and basically it causes apoptosis, which is cell death of bad white cells, the bad white cells that are causing a lymphoma or something
Yeah.
like that. Um, Other viral, obviously, complete plasma exchange is another possibility where you pull out somebody's. Actually, I was just reading. There's a guy who has who thinks he's figured out longevity, age
Uh,
longevity. I don't know
yeah.
if you've heard of
Brian
him.
Brian Johnson. Yeah.
Yeah.
I read about him.
Yeah. Um, but you can pull out, your plasma volume and everything that comes with it, and you can replace it with better plasma, fresher, healthier plasma. That's a plasma exchange.
Right.
That's another application for something like that. So there are many potential uses for that. I'm not sure that anti-aging is one of them, but
I wanted to ask you about that at the same. I mean, yeah. I mean, there's been a lot of hype in the news about that, and it goes back to sort of what you were talking about earlier. That look, we can't take everything out. I mean, there's a lot of stuff in there that's good, and maybe some of the things that are bad at a certain level, maybe when they come down or not. So this whole sort of pleiotropic nature of stuff that happens in us. But it gets me to that point about sort of the solution shortages that we had a couple months ago, and could you just say a couple words about that? Because I think that's important when it comes to some of these practices like total plasma exchange, where in certain cases, pathogenic cases, are very important. That we got to be kind of careful of sort of where we're going with that, because we don't want to have a shortage of albumin at any point, because there's a lot of other legitimate purposes. And if you could say a couple words about that, I appreciate it.
are you asking specifically about albumin, or are you thinking of the saline shortage?
Well, I think we had a shortage of many of those types of sort of replacement solutions a couple months ago, if I'm not mistaken. And maybe I'm talking out of turn here, but
Well,
um,
what comes to mind from my previous life is, post Hurricane Milton and Hurricane Helene,
okay,
um, those hurricanes had an effect on uh, North Carolina,
yeah,
which is where many, and that's my former life at Baxter, solutions are produced.
yeah,
In fact, 60 or 70% of the US market, I think, comes out of a facility there that was subject to, mudslides and some other things that took it out of operation. And that's a very tragic and serious situation. And so, yes, that did create shortages. And so I think the impact of that was really having to be very selective about where those solutions are used
hmm
and when they're used. Um, I'm not aware that that impacted our business specifically, but it certainly impacted the U.S. hospital business and, you know, elective surgical procedures and things like that.
I one other area I wanted to touch on with you, and this is, I sort of relayed this personal story to you a couple days ago when we were chatting, but sort of the area of palliative care and the use of some of these technologies. And I told you the example of my father-in-law, which, after he sort of exhausted standard of care and in his metastatic cancer journey, went into palliative care. And, I had never seen this before in terms of just the blood transfusions, but really, I don't know, I used the word rejuvenated him, but really, did for several months, sort of give him additional quality of life, additional progression-free survival from the perspective of, an outsider. And I would just love to get your perspective. I know there's, different rules in the United States in terms of, how we use these technologies and transfusions and so forth in palliative care versus hospice and end of life. But love to just get your own thoughts, perspectives on that particular area of application.
Yeah, gosh, Ira. Yeah, we did talk about that, and we have a lot in common there. Like you, my father had leukemia at the end of his life, probably the last year. And as a result of that, he became anemic. I mean, his red cells were not surviving. And so he actually had over 60 um, transfusions with red cells, with whole blood and red cells. And it's one of the reasons that I feel so committed to being a donor, because it did honestly extend his life, I would say, close to a year. Eventually, it became, a law of diminishing returns
Right.
as things progressed, but I'm really grateful that we had that extra year, and it's because people donate. And so again, I'm a big supporter of that. I think it's a very safe way to treat people without really a whole lot of side effects. And again, I'm going to put in a plug for all of your listeners to think about donating if and when you can. There's some reasons occasionally why you can't, but it's really important. It's you don't know when that type of therapy is going to come back around to your family, your friends, your neighbors. So, yeah, I'm a big fan of that, and I was very grateful. It was actually kind of shocking to me that somebody could go through that much blood, um, for such a long period of time. But it worked, and I was grateful to have an extra year.
Yeah, no, that's a wonderful story, and again, if there was infinity dollars that fell from the sky, I would love to see a lot of research focused on that particular segment because I think like in a lot of the areas that we've been talking about that we're just beginning to understand the applications. I think the that particular population there may be a lot left to discover on what can be done there. Per the palliative sort of hospice zone. So that's something of extreme interest.
Agree.
I'm glad you touched on it. Um,
Absolutely.
Jackie, what else should we be on the lookout for in terms of sort of the next generation of technologies? I mean, you mentioned photopheresis to us. You mentioned the ability to make these technologies smaller, more portable. What else, looking out on the horizon a bit that we might have not discussed yet, that you can mention that's not confidential, should we be looking at in terms of this space?
I guess so. This is Jackie's Jackie's vision
Wish list.
only.
Okay.
Yeah. Like I said, ge geographical expansion is really important. We have a great ability to serve, the Americas and Europe. There's a lot more out there in Asia in particular and Africa that's unaddressed. So I think that's definitely one thing that I hope to see in my lifetime.
Hmm.
The other thing, like I mentioned at the beginning, I've had so many neighbors and friends affected by rare diseases. Um,
Yep.
like I mentioned, Lou Gehrig's disease, that's one that it's just so brutal that I'm really curious to see if there's something we could do in that space. pancreatic cancer is another one. You get pancreatic cancer, it's just not good.
Yeah.
There's not a lot of hope there. So I personally want to go to the areas where there's very little hope right now. I think that's something that makes sense to address, and certainly would feel good to be able to address, whether it's geographic or whether it's just, an orphan disease situation that doesn't have a solution today.
And briefly, just say a couple words also about if in saying that the folks that are listening and going to be watching the show, if there are promising investigators out there that have decent ideas for how to scavenge an interesting type of cell or has an interesting biologic target, how do they go about collaborating with Terumo BCT? What are some of the ways that your organization works with the smaller investigators, the university labs, and so forth?
So there's a variety of ways. One is we have a business development group that looks for partnerships. We have the R&D team, me specifically, or anybody on my team, that people can reach out to directly with ideas. We have a clinical trial organization who, constantly partners with investigators, especially in areas like, let's say, Alzheimer's disease or leukemia, or so we do a lot of partnerships currently. I think one of the areas that we're looking at now from a partnership standpoint is, like you mentioned, are there molecules that can selectively target or remove bad guys that we could incorporate into our instrumentation in some way, so that we, achieve removal of something during a procedure. So absolutely, we have a person by the name of Robin Corcus, who leads our partnership area and strategy. And then, of course, myself, I'm always available. I sleep with my phones.
Well, you have quite a wide-ranging portfolio, and clearly the therapeutic opportunities are vast. Whether we're talking about scavenging bad guys, collecting good guys and modifying them with, as you were mentioning, making CAR T cells, gene therapies. It's a really amazing package. I wish you and the your team and the company the best with all of it, because there is clearly so much potential there for all aspects of therapeutic intervention. So really, an exciting time and discussion, and I love to see this technology take off as it has, and continue to. I look forward to continue watching your journey, and as you succeed in bringing these opportunities to more patients around the world. But really great stuff. Again, for everybody that's going to be listening to this episode of our show across the various podcast networks, or who are going to be joining us on the YouTube channel, again, you've been spending time with Dr. Jackie Kunzler, Senior Vice President, Research and Development, Terumo Blood and Cell Technologies, doing really amazing things, as they say, unlock the potential of blood, do more for patients than it does today. Jackie, again, I want to thank you for taking the time out of your schedule to talk to us for a little while about what you're up to. Obviously, thank you for everything you do. And as we like to say on our show, like you do, we want to create a better tomorrow. And I really appreciate the way you and your team create a better tomorrow for all of us. So thank you so much for that.
Thank you so much, Ira. I am so grateful to you for having this great discussion. I feel really very fortunate to be able to work in this field and for TBCT. And like you said, our goal, I think the goal of everybody who works in science and medicine is to make the world a better place. So thank you for giving me this opportunity.
Wonderful having you.